And the next talk is on transfer learning approach for aerosol particle scattering image classification by Skhathi and Mthembu from University of
Advanced. This is a test. Hi everyone, my name is Skhathi, and I will be talking to you about transfer learning and trying to use that in classifying aerosol particles using their forward scattering patterns. My presentation will be in this form, so I'll go through the project rationale, why we study this, why we care about forward scattering, why we care about machine learning. I'll look at particle physics and microstructure. I'll look at scattering experiments, effectively how my experiment works, the one from which I get data. I'll then look at said data. We'll go through some results. We'll chat about those results. I'll summarize this. It'll look exactly like this, but at the end. And then I'll open the floor for questioning. All on the same page. Groovy. Just before we carry on, I think it's important to note the size distributions that we're working on in aerosol science. So other people may be experts at different parts of this scale, but where my research is hyper interested is in the micrometer scale, because those are, I want to say micrometers, I mean very small micrometers, so hundreds of nanometers to the thousands, because those are comparable to the wavelengths of the laser that I use. Spoiler alert, there's a laser. All right, aerosol science has kind of a few disciplines. We've gone through them. I'm not going to belabor the point, so I'm just going to keep it pushing and go to the actual juice of this. Before we carry on, I'd like to go through a little bit of, I suppose, background on why it's important to care about forward scattering, why, and the kind of underlying principles that govern scattering and why we care about them. When there is light, in the beginning, when there is light impinging on a particle, there are a few things that the light can do. It can ignore the particle completely, so it's not involved, or it can undergo reflection, it can undergo refraction, or diffraction and absorption. Mostly it's diffraction. You can also get Raman or fluorescence, but I don't really care about that. And we can avoid that by controlling for the energy or the wavelength or the frequency or the mass of the light. I'm kidding. But anyway, the resultant image that you get from all of these interacting with each other, an image can be seen by placing some photosensitive thing here. And the resulting images that you get from that can tell you something about the interaction between the incident light and the particle that the light impinged on. The things that they can, this is not supposed to be here, but anyway, the things that they can tell you about this can be shaped. So you can learn about what the kind of shape of the particle you're looking at. I think that explains itself. You can look at the composition. Am I dealing with a water particle? Am I dealing with grains of sand or a combination of different materials? It can tell you something about the size. So is it a droplet of some radius relative to another? And it can tell you something about inclusions, what kind of particle we're dealing with, and what is inside, next to it, attached to it, stuck to it, etc. All these things are affected by these interactions, and there's a little bit of inference that we can do in order to know what kind of particles we're looking at. We've introduced a very fun camera on this side, and we've tracked these, we haven't tracked them, but like we see these interactions as a net kind of response, that these are the data that I am looking at. These are our forward scattering data. Before we carry on too fast, we also need to talk about my experiment. Now this is the kind of setup that we have for any scattering experiment. I used two machines, but I hadn't really used them. A lot of people took data readings. I just look at their data, so I don't really know too much about these machines, except that they're really awesome and they use lasers. But the basic premise is pretty much the same. You have some aerosol particles, normal to the direction of a laser. The laser then hits these particles, and those fun little things happen on this surface. There's a bit of a block there. There's reason I'm mentioning the block. We call this a beam stop. And we put it there so that whatever optical detectors we have aren't bombarded by a super high energy thing, a laser. We don't care about the central part. We care more about the particles that are the information encompassed by the scattered light. If you look at 1 and 2, these are two separate. There's a word for this, two like machines. It's not machines. There's a word I'm looking for that's escaping me right now. Anyway, that we use for our experiment. 1 is the airflow, so that is 1, and it's 1 here as well. 2 is the direction of the laser. We use a green laser because it's a really fun color, but also because we don't get Raman spectroscopy influences from that. So we get no absorption and re-emission of different energies, light, which is quite fun. Makes our experiment really simple as well. Now that we're all experts on this, I'm going to go through the actual project, which is the use of different neural networks on classifying these sport scattering images. I haven't shown you a single scattering image right now, so the suspense must be killing you. But my actual research involves using 3 different types of architectures. A bespoke one. Bespoke sounds really fun, but it just means I did that myself. There's also a pre-trained CNN. When I say pre-trained, as in, like, if you know about transfer learning, you use a model that's already been trained somewhere else. The idea of which is it's a lot better at recognizing stuff. I've got an analogy, a fun one. My niece, we're watching Rise of the Planets of the Apes, and she saw an ape for the first time, that ape that can speak, and it's really groovy. And she says, monkey. And we all just laugh at it because it was really hilarious. But like, she doesn't really know the difference between an orangutan, a monkey, or an ape. And we human beings that are a lot older than her do. So that's the kind of concept of transfer learning. You have somebody that can identify different genuses of animals, and you teach them how to identify one sort of like relational group a lot better. So now she knows the difference between an ape and an orangutan and a monkey. So that's what we're doing here. We're using some model that, like my niece, has already seen different kinds of animals, different kinds of things. Really belabored that point, but I just wanted to tell you guys that. And the last one is a pre-trained CNN. CNN stands for Convolutional Neural Network. It's right up there. I use the pre-trained one that has fine-tuning. The difference with this, or rather the addition, fine-tuning, is effectively another way of making sure that you have some training. So instead of just having a model that goes forward and backwards, like transferring information, moving things, fine-tuning does not actually change my weights. So it just changes the last bit of some model. It's a different way to do it, and we're trying to find the differences, see which one works better. Yeah, let's find the optimum model. It's a bit ahead of us. So these are the scattering images that we're looking at. I'm purposely giving you this because I know it's difficult to see, but it is also the exact images that we get from our 2 lab equipment, the SID, the SID3, and the PPD2. But we do some processing to it to make it to there. Anyway, we do some processing to the image so that we have the same kind of input going into the model, and the training requirements are fulfilled. So you want a certain number of images, etc., etc. The data to the for scattering images, where each pixel is effectively some intensity, and that intensity corresponds to a value on some 2D matrix. So the size of this corresponds to the shape of an input matrix. That is why we have this converted into something that looks a little bit more like a square. We actually do want our input data to look like a square. It makes it easier for me to do it. And these are the aerosol particles that we've collected for scattering images of. So there are different kinds of fibers. Do not ask me what these are. I do not know. We have that word that I shall forever refer to as BG, because I do not know how to pronounce it. There's salt, puffball spores, oval-bumin sucrose, beta mulberry pollen, and E. coli, PBS alpha, and unwashed. These are just aerosolization. Like if you see the same thing, but with different stuff attached to it, either pre or post, it's the same thing. It's just been aerosolized a little differently. We're trying to see if we can see the difference between those two. The model shows that we kind of can. All right, now I've recolored these so that you can actually see what they look like. So if you're waiting, but the processing we did was to black out the center. And for those of you who are a little more eagle-eyed, you might see that there it looks like I've blacked out around that image. So what I did was we call this a region of interest. So that's where all of our interesting scattering occurs. Because this is a forward scattering problem, you have to quantify how much forwardness is the forward scattering. Surprise, surprise, it's 25 degrees. So our detectors, our cameras effectively can only see from zero, not really zero because we block out the center. So from 8 degrees, like from 8 degrees to 25 degrees, and that is contained in that region that you look at. Anything else outside becomes a bit of an artifact. So I got rid of it by just blacking it out. When I say black it out, I mean I dropped the intensity of the pixel to 0 instead of having it as whatever it was. I did the same for the inside, because if you look at the previous image, you'll see this. This is an artifact of the beam stalk interacting with the beam or the laser beam itself. And that kind of remains very similar for each campaign that you do. So if I'm doing all my data today and I don't move the detector, I'll have the same artifact, which really sucks, because if you're taking data as a human being, you tend to do the same particles on one day, and then the next day something happens that changes the state of your lab, and you do it again. So your artifact might be different, and this is a place that your model can kind of get stuck, especially because it's such an easy thing to see. So blacking that out kind of makes sure that our model doesn't observe a local minimum in the form of that artifact. Ruby. Now we'll go to the results. This is a confusion matrix. I know there's a lot of stuff going on here. There are a lot of numbers in here. So a confusion matrix, for those of you who do not, is a measure of how confused your model is. The diagonal that you see kind of pronounced for being there from darker blue is effectively the thing that you want. So a perfect model will have a factor of 1 through the whole diagonals, which means that every particle has been properly classified 100% of the time. If I have a 0.5, it means that it's been classified 50% of the time, where the other 50% can be spread to anywhere along the row. It can be spread anywhere along there. I don't really want to go too much into this, but I just want you to look at the darkest ones relative to the not dark ones and where they lie on this. Like, make mental notes for yourself. I'm asking you to be engaged. Make mental notes for yourself to around where you're not getting nice classification. I'm going to say BG washed Sonatec big, BG Sonatec, and yeah, these are not giving what they need to give. But if you look at the rest, the diagonals seem pretty dark. So your model's doing something, except that BG wash is just not winning, and it never seems to win. In fact, all these three never seem to win, which can suggest that my model just cannot resolve between different kinds of that. So we can't see the difference between BG and BG. And you can see that E. coli, although not powerful, we're looking at 73. Does some work, but it's not brilliant. I'm going to move from this one. So this is just a comparison. I haven't told you exactly what I'm comparing with what, but this is the confusion matrix for 3 CNN models for the first quartile. You're seeing a quartile there. So what I did was I divided my data into 4 different quartiles, and I started removing the lower ones systematically. I first remove the first quartile so that I remove any data that don't have high enough intensity. Intensity just means the sum of all the pixels. So if they're not bright enough, you can't see them clearly, they're not involved in my analysis. I'm not going to tell you why I did that, just want you guys to pay attention. I did this again for the second quartile removal. That means not just the second, but the first and the second. It's cumulative. And I did it for the third quartile. And you can see some changes, but the same things are pretty much like observed. You've got the diagonals and you've got a lot of confusion over here with the BGs and the E. coli. I'm then going to go to another kind of comparison where I'm going to look at the untrained quartile comparison. That's quartile 1, 2, 3, and quartile 0. Quartile 0 is just like no removal of data at all. So I put all my data into the model and see how it goes. You can see that for quartile 0, like in my untrained, in my sort of OG model, quartile 0 is winning. So quartile 0 is that girl, like best results. But then you start going to your pre-trained models and it stops doing very well. Your quartile 2 is a little bit of a winner here. And we see quartile 2 doing really well once you edit your data, once you remove the low intensity images. And that might just be because you've got a lot of images that don't really belong to any class. They might be misfires, haven't really gone manually through the data to remove anything that we can see as not belonging there. That's a case for future work. But also for, that's what I wanted to communicate with these things. So, now this is the main thing I wanted to highlight you guys about. So I compared all 3 of them for the second quartile because that had the highest just general sort of accuracy from all the quartile comparisons. And I think that might be because I hadn't removed so much data that my training started to become compromised. Garbage in, garbage out. But also I had enough data just like to have a really nice training run, because once I remove quartile 3, I don't have a lot of data, and CNNs are notoriously data hungry. Back to this image though. The untrained did the poorest in these. The pre-trained did pretty well, and the fine-tuned did pretty well as well. Like mostly, it did a lot better. Mostly, my accuracies were between 60 and 75%, so I never got anything above 75%. I'm also dealing with like a multi-class problem. So what is accuracy? Do I just average all of them? Some things aren't doing very well. If you had an eagle eye, you would have seen that fiber is always right up there. Fiber is really easy to identify, but I had my data, I had my fiber not consolidated as fiber, but in different sort of classes by themselves. So I had gypsum, I had SW, etc. And that was just terrible. Fiber was the worst performing. So I'm saying that because I think there is some hope for BG and BG washed with some pre-processing and some aerosolized step, because I think my model is just agnostic to the differences between them. So if I unify them, maybe I'll have a lot better model. But this is future work. Anyway, we've seen this. And yeah, just to go back to the model performance again, there was places to rather improve on the model are just like general performance and misclassification, which I've kind of touched upon, the consolidating of my BG into the same class, irrespective of the pre-processing and the aerosolization that it underwent. Also the quartile selection. The reason I selected my quartiles that way is because I had dark data. Now I need to remove it and make sure that my data look great. So that's another step. Overfitting, something about too many parameters, but for the sake of time, I think that it is worth looking at other ways of trying to remove that central beam instead of just blacking it out, because it increases parameters and it makes this model tend to overfit. And that's about it, really. That's about it. You guys remember what I talked about. Thank you.
little over time now, so maybe if anybody has a quick question,
No one.
if not, we'll resolve the circle of confusion after the break.
Fabulous. Thank you, guys.
Thank you.